Good morning, and thank you, Donald, for the invitation to speak today, and thank you to the organisers. It's been a fantastic event, and I've learned a lot as well listening to the talks today. So the presentation I'm going to give is my personal views based on my experience as an academic researcher. So I work at the University of Southampton. This is the building I'm based in at the hospital site. So if you ever find yourself on the south coast of the UK, please do pay us a visit. And it's a great privilege for me to work in a department with a range of colleagues with expertise in the nutrition sciences. Nutrition and immune function is something which has gained a great deal of interest since the COVID pandemic. Before COVID, my research was focused on prevention of allergy, but post-COVID, the public, commercial and academic interest in nutrition and immune function has grown significantly. This means that I have some declarations of interest to make, both in terms of professional societies that I have responsibilities in, commercial organisations that I deliver talks for, and also other research funding that I receive. But what I want to talk to you in my presentation today about is the role that nutrition can have upon your immune system. And I would separate this into three main parts: the energy that's required by your immune cells, the specific ingredients they need to do their functions, and those important interactions with the microbiome, which we heard about yesterday in the talks. Over my career, most of my research has been in the field of omega-3 fatty acids or prebiotics and probiotics, so those will be my focus today, but of course those aren't the only nutrients that affect the immune system. And as I mentioned a moment ago, I started my research career looking at allergy prevention, but since that time I've started to do work on the role of inflammation, particularly in those living with obesity, the role of aging, and post-COVID, the effect of nutrition on infectious disease. And this is one of my favourite cartoons, which I'll come back to as we start talking about those gut-immune interactions. But first of all, if we think about our immune system, one of the things I mentioned is that your immune system requires energy to do its function. I think this short video illustrates that nicely. We've got a neutrophil here chasing down a bacteria in order to engulf it and then destroy it. So I want you to reflect on how much energy that cell requires to do that process, the properties of the cell membrane that are required to have that flexibility to move where required, and then the specific functional components that may be required to generate those reactive oxygen species to destroy and target that bacteria. So when our immune system is active in this way, we have an increased energy demand. So if you're suffering from an infection, there's been some estimates made that for every degree of fever you have, you need an extra 100 calories a day, for example. That's a very rough estimate, but we all know that experience of if we've been unwell or we're recovering from injury or surgery, we may have some weight loss over that time if our appetite declines or we have these additional energy requirements. And so we can either meet those requirements from our dietary supply, but if we don't have a good appetite or we don't have a good dietary supply available to us, we will start to draw upon our body stores, and that can cause some further complications. I also mentioned the role of nutrition in specific building blocks for your immune system. So, for example, vitamins and minerals are classic examples of essential nutrients, and the reason they're essential is that your cells, enzymes, for example, require them as components in their structure in order to function. So the role of vitamins as cofactors, for example, in enzyme function. And this can include both macronutrients, so specific amino acids like arginine required to generate nitric oxide to target those bacteria. And in human health, we're very well aware of the important effect of vitamin A and zinc on immune function. So because vitamin A and zinc have this key role in cell division, if you don't have enough of that, your immune cells cannot proliferate. And the outcome that we see of that is increased mortality amongst children who are living in areas of deficiency. And many millions of childhood deaths can be attributed to the entirely preventable problem of vitamin A and zinc deficiency each year. So there are various immune tests we can do, and there's some debate and discussion about what are the most valid tests of immune function to do. One which is highly regarded is vaccine responses. We've seen data from that presented today. Here's another example of one which is deemed to be highly clinically relevant, which is this delayed type hypersensitivity reaction, where you expose someone to an antigen, and if their immune function is working well, they should get a nice large red wheal afterwards. But if they're deficient, their immune system may not respond appropriately to that. And I'd like you to think about what that means. So, for example, if you get a splinter in your finger will become red, hot, swollen, painful, and possibly you'll lose function of being able to press your mouse key. But that inflammation at the right place at the right time is positive because it prevents the bacteria from tracking into your bloodstream and causing you to get sepsis. Okay, so inflammation in the right place at the right time is a good thing to have, but chronic low-level inflammation would be a problem. The interactions between the microbiome and our immune system are numerous and complex, and so I'll try to highlight a few of the key areas that I think are important for our talk today. So broadly, those of us working in nutrition and the gut microbiome space think about the balance between what we might describe as beneficial bacteria in our gut versus those that we might consider to be pathogens or problems. And what we're trying to do through diet, and I'll talk through these various components shortly, is to promote the growth and development of beneficial bacteria in order that they can both compete with pathogens, so they can physically occupy the space. They can make these important metabolites, short-chain fatty acids we've heard mentioned a few times today. Those are derived from the action of these bacteria on carbohydrates resources within the gut. You can also make branch-chain fatty acids if you have a high-protein diet. There is still lipid within feces, so these bacteria are not just metabolizing carbohydrate, but that's one of their preferred sources. These in themselves can acidify the gut contents, and we saw some of that data on Clostridium species yesterday and how that affects differential growth of those. And these short-chain fatty acids can also provide energy to your gut and interact with those immune cells directly. And so by this whole range of interactions, what we're hoping is that through diet, we can modify the type of bacteria in the gut and improve both local immunity, but also to have these whole body effects. Now, I want you to reflect on the constant and massive antigenic stimulation we all give ourselves each day. Each of us eats probably 1 to 2 kilos of food a day, and our body has to surveil that and make sure that it's not containing pathogens. If we had the misfortune of eating something contaminated with salmonella, our immune system would activate a strong and protective response to eliminate that from us quickly. So in that circumstance, we want this vigorous, rapid response to infection. But each day we're also exposed to food proteins and commensal or harmless bacteria within our gut. And we don't want our immune system to overreact to those. If we're overreacting to food proteins or other environmental allergens, we might be developing food allergies, which can be life-threatening. If we're overreacting to friendly bacteria within our gut, we can develop conditions like inflammatory bowel diseases. So our immune system has to walk this careful tightrope of being strong and effective, but not overreacting to things in our environment. And one thing that nutritionists sometimes can be prone to is taking a rather reductionist approach. So let's say, for example, I observe that people who eat walnuts frequently in a Mediterranean region have reduced risk of cardiovascular disease. Some people might then say, okay, well, what is the component of walnuts which is causing this? And I might suggest it's probably the alpha-linolenic acid found within the walnuts. And so people like me will do a trial of giving people capsules with that specific component, using it like a pharmacology trial, an additional medicine that we're trying to give you. But one of the things I want to remind you all is that the food we eat is not just about the nutrients in it. It's more than just the sum of its nutrient parts. Eating walnuts contains a whole range of nutrients, as well as those fatty acids, I think, have those cardiovascular disease prevention properties. It's also a source of protein, phytochemicals, fiber, various vitamins and minerals. And so I would encourage us to think about avoiding that reductionist approach and thinking about the food that we eat as a whole. And that means including some of the cultural activities that are related to the foods we eat, the psychological and emotional aspects of the food choices that we make, and those wider social contexts, those influences of policy and government upon the food choices that we choose to make or that we have the ability to make at all because of what's available to us. So here's an example of some of the varied factors that affect your food choices now. So let's say you're thinking about what to have at the snack time out there. You may have some perception of what smells nice as you're going out, and you can smell the delicious pastries out there. You may have some factors in your personality. Your experience yesterday with the pastries being very nice may have made you more confident to select them again today. Their relative availability, there was no need for us to pay for any of this, so we could choose anything we wanted at any time. But there are limitations in what's available to us. If I wanted to eat a kiwi, there was no kiwi yesterday, right? So although I have a degree of choice, I don't have full choice to do that. And the thing that I often say to my students is that my students will often say, well, eating healthily is very easy, right? People just need to be disciplined and eat right, and then all the problems of obesity will be solved. And the example I like to give is to imagine you're sitting in your living room and you want to eat a banana. What are the steps you would need to take to get that banana? And for me, it would be to get in my car, drive 3 miles, go to the supermarket, buy a banana, return 3 miles in my car and go home. If I wanted to get a chocolate bar, I could leave my front door, walk 50 meters to the corner shop, buy a chocolate bar and return home. And so you can think about the accessibility I have to access a banana because I own a car, perhaps have the luxury of time to do that. I can afford the fuel to put in it. Makes me in a different situation than, for example, my teenage daughter, who doesn't have the accessibility to do that, but can take herself to and from the corner shop. So I encourage you to think about all those effects of food. And sadly, we live in interesting times where there are further pricing constraints being placed upon all of us. We've probably all experienced this in our home shopping basket so far. This is data showing that really just over the last year, the dramatic increase there has been in household shopping within the UK. And not only will this affect food choices for humans and what we choose to have, I think it will also be likely to affect the food choices that pet owners make for their companion animals as well. And so there could also be some challenges around those aspects of food choice. But one thing I'd like to share with you is that malnutrition is a significant public health problem even within countries such as the UK, and that's something that people sometimes don't think about. Malnutrition accounts for 15% of the spend of the healthcare budget in the UK, and it's important for us to note that malnutrition can relate to undernutrition, either of total energy or of a specific nutrient. So most older adults admitted to hospital in the UK are malnourished on admission when they go in, and during their admission become even more malnourished. Okay, so that's the sad picture that we have in the UK. But overnutrition and obesity is also a form of malnutrition as well. And sadly, I think we have a tidal wave coming towards us of obesity arising from some of the complex social changes that occurred during the COVID pandemic. So this data here is the rates of childhood obesity in the UK, measured in children who are in what we call reception, which you might refer to as kindergarten, or children who are 4 to 5 years of age. On entry to school, all children in the UK are weighed and measured, which provides fantastic data for people like me working in the nutrition space. And what I'd like to highlight to you is that since 2006, the levels of severe obesity had been unchanged, and we viewed that as a problem, right? That we weren't able to nudge these numbers down. But what we saw in COVID was a shocking increase, doubling the rate of children living with severe obesity, significant increases in those with any kind of obesity or overweight. In such a short space of time, this was really quite horrifying. And while the figures now are starting to move in the right direction, even this increase to 2.9 from what was a historic steady state of 2.4 is deeply concerning. And we will have to wait to see the data. These children will be weighed and measured again when they leave primary school at the age of 11. We'll have to see whether that effect persists. But I fear there is a tidal wave coming towards us as these young people become adults, and we've started to talk a little bit about the developmental origins of health and disease and these impacts here. Now, quite often when we're talking about childhood obesity, people will say, if you look at the family as a whole, perhaps there's a genetic factor involved in this, perhaps there's some genes going. I've heard some comments about perhaps this particularly inherited from the mother because the mother's body composition seems to reflect what's happening in the child, and perhaps in the mitochondria sessions this afternoon we might hear some explanations for that. But perhaps all of us have also noticed occasionally that sometimes the family pet also has the same characteristics, and so that can't be explained by genetics, but perhaps there's some other social forces acting upon that family as well. And so I think something important for us to consider there. Now, doing nutrition research is challenging because I can't always blind you to an intervention. Let's say I wanted to randomize half the room to fish oil and half to control. Half of you will open your capsules and they'll smell like fish, and the other half they won't. So it's difficult for me to blind you. In finding out what your background dietary intake is like, that's also a challenge, because even if you answer my questionnaire honestly and say, so I had a banana in the break just now, the bananas outside, I would say, are mainly a type 3, which is going to give you about 15 grams of resistant starch. But if we left those bananas for a few days, you'd have about 15 grams of sugar in your type 7 banana. So even if you know I ate a banana, it's not enough information for you to know what I'm eating. How acceptable is it, the type of study that I'm trying to do? If I ask all of you to now go on a fasting diet and not eat anything until tomorrow morning, we'd all feel very sad at the dinner this evening. So sometimes there can be challenges of compliance, and we end up with self-selecting groups who are willing to do the things that we're asking them to do. So there are some significant challenges with doing nutrition research, and sadly that means that not all nutrition research is robust science. I hope, of course, that the work that I do is robust science, but there are challenges with that. So I mentioned some of those specific building blocks of nutrients in the immune system. Here's a list of some examples of them. But one thing I wanted to highlight to you is that in terms of food labeling for humans, there are a limited number for which claims can be made, which say they contribute to normal function of the immune system. This is quite a modest claim, saying that this is something which has a role in your immune system working normally. It's not suggesting that taking a higher intake of that is going to improve your immune system further. And so you may have, in your experience, seen products which describe themselves as immune boosting or superfoods, for example. And so we need to take care with the kind of language that we're using in nutrition. Well, let me move on to tell you a little bit about some of the immune-related work that I've done in nutrition. So one area I'm particularly interested in is omega-3 fatty acids. So in the human diet, there are two essential fatty acids, alpha-linolenic acid and linoleic acid. Now, we have a series of enzymes which allow us to convert these essential fatty acids into the longer chain products, which can be arachidonic acid or EPA and DHA, these long chain, highly unsaturated fatty acids. And cast your mind back to that cell I showed you at the beginning with the highly flexible cell membrane. These fatty acids, these long chain fatty acids, allow that flexibility there as well. So they have structural roles, signaling roles, as well as their energy providing roles. We've also touched a little bit in the last few days about potential sex differences in either rates of diseases or responses to treatment. And this is another area where women are much better able to do this conversion with men. And there's some speculation that that's in order to provide nutrients to a potential pregnancy, that we have that ability to do that. But we have this pathway which allows us to do this process. And one of the reasons this is important is that these fatty acids are precursors to important immune signaling molecules. So for arachidonic acid, that's used to generate the prostaglandins and the leukotrienes, which are classic markers of inflammation and allergic diseases such as asthma. Now, if we have a diet which instead provides us plenty of EPA by eating oily fish, in the UK, we're recommended to eat oily fish once a week. The median intake of oily fish in the UK is 0 grams per day. So most people in the UK never eat oily fish, despite that being the recommendation. But if they were to eat EPA, that would competitively inhibit this effect on arachidonic acid and instead would produce an alternative series of these mediators, which are broadly considered to be less inflammatory. Now, there was a hypothesis that those people who had the highest intakes of these omega-3 fatty acids, such as those in Inuit communities, that was seen to be associated with them having much lower risk of allergic disease. And because of that mechanism we understand with prostaglandins, this is a plausible explanation. So then some more detailed observational studies were done, particularly in Australia, where they were able to attribute some incidences of childhood allergy to those who had a very high intake of margarines and spreads that had the omega-6 type of fatty acid, and a protective effect of eating oily fish amongst those children. But one of the things I would encourage you to remember is that this is observational data, and that children who have regular consumption of fresh oily fish, I might suggest, are likely to be from a different socioeconomic class than those children who do not, from a different cultural background than those children who do not. So we have to take care with these food associations and what they actually mean. So then intervention studies were attempted, and this developmental origins of health and disease was talked about. So as asthma and allergy develops in early life, many of these studies have provided supplements either during pregnancy, during lactation, in those early days of life, to see whether that can influence immune function. So a number of these studies have been done both in Australian institutions but also at the University of Southampton, where a study was providing salmon to feed the family twice a week to people in that study, the salmon in pregnancy study. And broadly, this still remains a controversial area. So despite there being a clear mechanism, clear observational evidence, when it comes to intervention studies, it's hard to translate that across. We do see some benefits in at-risk populations. So women and parents who have established allergic disease, it seems to reduce the risk of their children developing allergic disease. But it's not beneficial to all. There's no evidence that it can be used to treat allergy amongst people who already have it. And so the current view is that we have to personalise those recommendations, and there can be some differences in the people who benefit and those who don't. I mentioned earlier that the interest in sex differences that I have, and I have a current research project looking at adults with asthma and seeing whether sex hormones have an effect on their ability to metabolise these fatty acids. But let's move away from omega-3s and towards the microbiome. The links between the microbiome and allergy have been discussed for some time, even before we knew it was the microbiome. So we talked about the hygiene hypothesis for a long time, that our improved cleanliness and living standards was linked to this increase in allergic disease that we were seeing. And certainly studies have identified that things like hygiene practices, the mode of birth, whether someone's delivered by cesarean or vaginal birth, will affect their risk of allergy later. And so there are a wide range of environmental exposures which are likely to link to our microbiome, one of which is pet ownership, being significantly protective against children developing allergic disease. But also we heard yesterday about the role of antibiotics. And as a nutritionist, we're very interested in the timing and the type of foods that are introduced and when to children. One thing I would like to say about food allergy is that there is some evidence of a modifiable nature to that, because some children will outgrow their food allergy, whereas others it will remain a lifelong allergy. I'm still waiting to outgrow my dog allergy. That hasn't happened, sadly. Hopefully one day I will outgrow that. But what we can see is these changes in gene methylation of various immune components. So these are cytokines involved classically in, for example, allergic disease. And we see that those who are outgrowing their food allergy are starting to move closer to what we see in the healthy control group. So this suggests that our environment is able to modify these things, and that environment could be other exposures to, for example, pollution in the environment, but diet and lifestyle are likely to be components of this. Now, I attempted with some colleagues at ILSI Europe to try and understand these things that could be promoting food allergy, and it's a complex picture, and I just wanted to show you this image to say that as a nutritionist, I need to work closely with colleagues who have expertise in genomics, the microbiome, epigenetics, in technological development, in order to get a full handle of what's happening, and that does present some challenges in doing this kind of work. But what about where the evidence is quite clear? So in inflammation and omega-3 fatty acids, there's a great deal of high quality evidence suggesting that fish oil can prevent this kind of inflammatory disease or treat it indeed. I would point out that these studies tend to use very high doses of omega-3. One piece of salmon probably has about 2.2 grams of EPA and DHA, so you'd have to be eating salmon every day plus to achieve this. So perhaps this is more of a pharmacological effect. But these studies of rheumatoid arthritis found that it reduced stiffness, the number of sore joints, the requirement for other medications such as non-steroidal anti-inflammatory drugs. So very convincing evidence to me from this meta-analysis. Yet still there are challenges for us in translating that to policy. In the UK, for example, clinicians are advised to inform adults with rheumatoid arthritis that there is no strong evidence that their arthritis will benefit from changing their diet. I disagree with that. They do say that adults could be encouraged to consume more fish, but given that no adults or the median intake is currently 0, I think that's quite a high expectation to think that just mentioning fish is going to be enough. So there are some challenges in these policy translation. But also, I don't want you to feel that omega-3 is the solution to all inflammatory diseases. There's a mixed picture of evidence across other inflammatory conditions, Alzheimer's, inflammatory bowel disease, cardiovascular disease, and I do feel it's controversial sometimes, the messaging that we see in the nutrition space about anti-inflammatory foods or pro-inflammatory foods. I think that can be a controversial area. The other thing I would like to emphasize that it is possible to do harm through nutrition. And so we do need to take care and be aware that there can be two-tailed effects of the interventions that we give. So I did this book chapter review looking at animal models of omega-3 fatty acids during pregnancy and the effect they had on inflammation within the brain. And so there was a number of advantages in these animal model systems of improvements to kind of measures of cognitive function and also resistance to damage from those challenges. But there was also evidence that it impaired fetal brain growth, the rates of maturation of the fetal brain, and the auditory development of the offspring. So we do need to take care that the same nutrient can have different effects on different systems as well. So a note of caution with nutrition studies. Now, I showed you this cartoon at the beginning of an immune cell reaching into the gut lumen to sample what's there to inform other immune cells. But of course, the picture is a little bit more complex in real life. We have these specialized areas called Peyer's patches. Here we can see this dendritic cell sampling the gut contents. And so this is a two-way interaction. The gut contents being taken up by these specialized areas of immune tissue, but also our immune cells secreting immune components into the gut lumen. So this is something which is a bidirectional interaction, and so a complex one for us to study. In terms of diet, one of the interventions we can do is to provide probiotics, which are the microorganisms directly. Those tend to be bifidobacteria or lactobacilli species, and so we provide those in the diet, hoping that they will either colonize or send signals to immune cells which are positive. We can also provide prebiotics. These tend to be indigestible fibers like fructooligosaccharides or galactooligosaccharides, specifically chosen to promote the growth of a friendly bacteria in our gut. And we can also combine those together. So we could provide a probiotic together with a prebiotic, and we call that a synbiotic. And they may be exerting separate but important effects, or they may be working synergistically, where the prebiotic is selected to specifically promote the growth of that microorganism. Now, in terms of the evidence for probiotics on health, I think the effect on gut health is very clear, and that's quite easy for us to understand, I think. If you are traveling somewhere where you're at risk of food poisoning, having a nice layer of friendly bacteria in your gut will prevent something like salmonella having the opportunity to adhere and cause you an infection. So people who are experiencing diarrhea, taking probiotics can reduce how long that lasts for, and broadly are a safe and acceptable treatment for people experiencing that. So antibiotic administration associated diarrhea can be prevented by co-administration of probiotics, for example, and I think the evidence for that now is quite clear and strong. But how about what's happening with aging? We see changes in the type of species of bacteria we have in our gut with age, and that reflects changes in our diet as we move from an exclusively milk-based diet towards making our own food choices and the quality of that, and then perhaps in later life having other challenges which are affecting our food choices. This illustration here is one designed to get students thinking about how someone with mobility challenges may have difficulty feeding themselves, and I don't know if you can see on the image, but his teeth are actually in the glass on the table here, so he would have some additional challenges there. And we know that the gut microbiota changes significantly with aging, but this also depends on the environment that you're in, and I would suggest the pattern of diet that's being consumed. So people who are older but living in the community have a much higher diversity of their gut microbiome than those who are in long-stay residential care. And so that may reflect changes in their exposures. Perhaps they're not going in and out of the home so often. Perhaps their diet is repetitive and stable, and so there can be some significant changes in what's happening with the gut microbiota, in addition to those changes which happen with age. The other thing I think is important for us to be aware of is that not all probiotics are the same. So I mentioned that they tend to be species of Bifidobacterium or Lactobacilli, and you can see that these have quite different effects on this particular measure of immune function. But the other thing that's quite striking is that they also have varied effects depending on whether you're using a young person's immune cells or an older person's immune cells. And so when we think about probiotics, one of the challenges we have is we can't think about them just as one big homogeneous group. We have to know what genus, species, strain of probiotic we have, and we also need to take care with what is our target population, which I think is something that Graham mentioned earlier. We need to take care that we're applying the findings that we have to the population the study was based on. Now, is there evidence that taking probiotics can prevent acute upper respiratory tract infections, cold and flu-like illness? I would suggest yes. This systematic review identified that they significantly reduced the incidence of people having a respiratory tract infection, the use of antibiotics, missed school days. But here we come back to that challenge we have in nutrition research, where they reported that there was a high level of heterogeneity of the data and that the evidence was considered low or very low quality. So doing high quality nutrition research is something that we really need now. But this is the picture that we're getting from studies. This is an example of a study I did of a symbiotic while I was at the University of Reading. So we were providing a symbiotic before people had their flu vaccination and for the 4 weeks afterwards to see if that had an effect on their response to flu vaccination. And Donald introduced us to these T-Rex earlier, and so I just wanted to highlight that this marker of aging of the immune system, here we have our younger adults who have much higher concentrations of these T-Rex in their T cells compared to our older adults, but there are some older adults with an immune system as good as the average young person, and there are some young people with an immune system as poor as the average older adult. So there's a great deal of variability in human studies which can pose a challenge. But one of the things we identified in this study was that although the probiotic didn't have any significant effect on outcomes, someone's relative markers of immune age were significantly associated with that. So people who had older looking immune cells were much less likely to respond appropriately to the vaccination, and I think that supports what we heard in the previous talk. Now, in terms of the evidence that probiotics can affect immune cell function, I would suggest that the evidence is very strong and clear regarding the positive effect they have on natural killer cell function. This is a recent meta-analysis which has been done on this topic, and again, a number of studies providing supplements for between 3 and 12 weeks found that it significantly increased natural killer cell activity, but again, we have a small number of low-quality studies with a large amount of variation between them. So a challenge for us in the nutrition research community. I mentioned earlier about those short-chain fatty acids, which can acidify the gut contents and change the pattern of bacteria which grow there. These can also have direct influences on immune cells. We heard in the talks yesterday about acetate, propionate, and butyrate. These can act upon receptors on the cell surface. They can also act upon important signaling molecules in inflammatory processes, and there's some suggestion that this can affect things such as clotting, atherosclerosis, other types of inflammatory diseases. So there's a great deal of interest in how these short-chain fatty acids might have this effect. And I mentioned probiotics, prebiotics, and symbiotics, but you may also see postbiotics, which is the dietary provision of these short-chain fatty acids. They need to be incorporated into a food matrix to allow them to be delivered to the large intestine rather than just absorbed immediately. And you may also have heard the term psychobiotic, which is a probiotic designed to have effects upon mental health arising from these interactions with the gut. One thing I would encourage us all to think about is what outcome measures matter to us most when we're doing studies. So here's an example study done in Chile. They had older adults and they were providing them with a mixed nutrient supplement. It had various antioxidants, minerals, lipids, and this prebiotic. And it was a flu vaccination study, so they were looking at vaccination responses. So this is the individuals at baseline and these days after vaccination. And broadly, to be protected against the flu, you need to cross this threshold here. So what we can see is that our adults gained protection after having the vaccination, and that this declined a little while after vaccination, but they were still protected. And we can see that there was a significant effect of this nutritional supplement there. But to be honest, this isn't super powerful, right? The control group was also protected against the flu. Does it matter if I say to you, your antibody titer is now 200 instead of 100, if 100 is already good? So is that meaningful to us? But something I might suggest would be more meaningful to us is this is the data they collected on people's self-reported days of cold and flu-like illness. And in the control group, there were a number of people who had some weeks of cold and flu-like illness over that winter period. And on average, the median was around 3 days of cold and flu-like illness over a winter period. But amongst those in the receiving the nutrient supplement, the majority of people reported no cold and flu-like symptoms over that entire winter period. So you can think for yourself, if I said to you, drink this every day and you'll have no cold and flu this winter, that would be quite meaningful to you, wouldn't it? There are challenges with these types of measures. How do we use this subjective data from individuals? How good are people at compliance? And I would suggest poor from my latest study. So we need to make sure that people are really engaged with this process. But I think this also allows us to build a case to policymakers. So they modelled if probiotics were able to prevent respiratory tract infections in the way that the literature suggests, what economic impact would that have? And in this model population of 50,000 people, it would reduce the number of courses of antibiotics by some hundreds, but of the greatest economic impact is the effect on sick days that individuals would take from work. And that's been estimated to save a significant sum for both the health services in terms of those periods of illness, but also for society as a whole as a result of those increased working capacity. So I mentioned that the evidence for natural killer cell activity was good, but one of the challenges we have is getting nutrition researchers to work with immunologists more closely. So of the studies that I reviewed, only 2 of them looking at probiotics in aging looked at any marker of immunosenescence. One of those was me, and the other one was someone else. And also in terms of study design, unfortunately in nutrition research, there are a number of single-blind, open-labeled, unrandomized or uncontrolled trials being published. So this is a challenge for us. But despite that, I would say that there is growing evidence of this role of probiotics in helping to support those who are aging. Some new concepts I would like to introduce you to, which I think we've been touched on a little bit yesterday, is this concept of increased gut permeability. This is the type of cartoon we see with a beautiful gut lining with a lovely mucus layer, and perhaps if this becomes damaged, allowing these microbial exposures to pass across and interact with our immune cells. As a nutritionist, the thing I would like you to remember is it's not just the bacteria that will be passing across, there'll also be nutrient transfer, and some of those nutrients can have direct effects upon immune cells. Historically, we thought of this permeability relating to inflammatory bowel diseases, but now we're starting to see evidence that it's associated with a number of non-communicable diseases like obesity, non-alcoholic fatty liver disease, for example. And when I was a student, we were mainly taught about peptide antigen presentation, but of course now we know there's also presentation of lipid molecules, and for me, I'm particularly interested in these mate cells, which are mucosal associated invariant T cells, which respond specifically to vitamin metabolites which arise from the gut microbiome. So a key cell linking what's happening in our gut microbiome to our immune function. My current work is looking at the role of phytochemicals on immune function. So as well as fruits and vegetables being delicious and a source of fiber and energy, the unique compounds that give them their colors and flavors are phytochemicals, and a number of these have now been identified to have anti-inflammatory or antioxidant properties and to link with prevention of respiratory tract infections or influenza. So I have a current study which is looking at two of these, taxipholin, which is found naturally in apple skins, and ergothionine, found in porcini mushrooms. So watch this space and I will update you on whether that had any effect on healthy older adults. These were adults aged 50 to 65. We heard yesterday some fantastic talks talking about bile acid signatures, so I would encourage you to think about those potential links of diet with bile acid signatures. I've mentioned the vitamin role that some of our gut bacteria can have, and there can also be specific bacteria associated with risk. Sulfate-reducing bacteria, for example, linked to GI disorders like ulcerative colitis. And we heard some great talks yesterday about the gut-brain axis. I might call it the gut-immune-brain axis, but of course things are even more complex than that. Adipose tissue contains immune cells and so is providing inflammatory signals for those living with obesity. We have the important role of the liver, very rich in immune cells, the first place that your digestive tract blood passes through. I focused on this link between the guts and things like asthma and allergic disease, but I have colleagues working on the skin microbiome and colleagues very interested in the effect of the microbiome on cognitive function. So I hope I've persuaded you that nutrition is important for immune function, both in terms of energy, building blocks, and microbiome interactions. I've shared some of the research with you on allergy, inflammation, aging, and infectious disease, and I hope that I've highlighted to you what I think are some of the emerging areas that we're going to see over the coming years. And I want you to bear in mind the important role that other organs have in immune function as well. So I'll just finish by acknowledging the fantastic wider team of colleagues and students who also contribute to the work that I do within my group, and thank you for your attention today. Thank you.